You know, in science, sometimes the biggest discoveries come down to the tiniest details. That's why today, we're going to talk about a true unsung hero of the modern lab, the micropipette, or as some call it, the sampler. It might look simple, but with this tool, precision is absolutely everything.
So let me ask you something. No matter what field you're in, genetics, biochem, you name it, what's that one little thing that can totally make or break your entire experiment?
Believe it or not, the answer is the precise transfer of incredibly tiny amounts of liquid. I mean, think about it. A massive, groundbreaking discovery could literally depend on getting one single drop from here to there perfectly, and this is the tool that makes that happen.
All right, so to really get you comfortable with this tool, we're going to cover 5 main things. First, why it's the hero of lab precision. Then we'll get into the different types and their key parts. After that, the core technique itself, how to actually use it. Then I've got some pro tips for you, and we'll wrap up with maintenance and calibration.
Okay, let's dive right into our first section. Before we talk about how to use it, we need to be really clear on what a micropipette is and why it's so important.
So what's its job? Super simple, really. A sampler is designed to do one thing perfectly: move tiny, super precise amounts of liquid. We're talking volumes as small as 1/10 of a microliter. The whole point is to make sure that the number you see on the display is the exact amount you move. And here's the other crucial part. You use a fresh, disposable tip for every single sample. That makes it our best weapon against cross-contamination, which, as you know, can ruin an entire experiment.
Okay, so now we know what it's for. Let's kind of look under the hood a bit. We're going to check out the different kinds you'll see in a lab and break down the main parts. First off, these are not a one-size-fits-all kind of tool. You've got your single-channel pipettes. Those are the workhorses for everyday tasks like setting up a PCR. Then you have the multi-channel ones with 8 or even 12 tips, which are lifesavers for stuff like 96-well ELISA plates. Some are fixed volume for when you're doing the same thing over and over. But the ones you'll see most often are the adjustable, variable volume kind. And the way they work can be different too. Most use air displacement, which is great for regular aqueous solutions. But for trickier stuff, like thick or volatile liquids, you'll need a positive displacement pipette.
And you know what's really cool? The design is so simple but so effective. You've got the plunger right on top, which controls everything, the digital display so you know exactly what volume you set. Down at the bottom, there's the tip cone, which is key for getting a perfect seal. And of course, the tip ejector for getting rid of used tips quickly and safely. That's it. Just a few key parts all working together to give you amazing precision.
All right, so knowing the parts is great, but using them the right way, that's what really matters. So let's get into the most important section, the core technique. Honestly, this is what separates good results from truly great, reliable ones. Okay, the number one thing you have to master is the plunger's 2 stops. It's a 2-step process. To suck up the liquid, you press the plunger down to that first stop. Then, dip the tip just a couple of millimeters into your liquid and slowly let the plunger come back up. Got it? Now, to get the liquid out, you move over to your new tube, touch the tip against the inside wall, that little touch helps a lot, and then you press the plunger down smoothly, past that first stop, all the way to the second stop. That extra push at the end, that's the blowout, and it makes sure every single bit of your sample gets out of the tip.
Okay, so once you've got that basic technique down pat, there are a few little tricks that can take your accuracy to a whole new level. Let's talk about some pro tips.
This one is huge. Always, always, always use the smallest pipette that can handle the volume you need. Why? Because a pipette is most accurate in the top half of its range. So let's say you need 15 microliters. You could use a P200, but you'll get way more accurate results if you use a P20. Choosing the right tool for the job is honestly step 1 for getting precise results.
So what about those tricky liquids? You know, like glycerol that's really thick, or things that evaporate easily. The standard technique we just talked about, well, it doesn't work so well for those. That's where reverse pipetting comes in. It's kind of backward. You start by pressing all the way down to the second stop to pull in a little extra liquid. Then, when you dispense, you only press down to the first stop. You're intentionally leaving a little bit behind in the tip. It's a really clever way to make sure that the sticky film that clings to the inside doesn't mess up your measurement. Okay, let's do a quick fire round of do's and don'ts. Do, hold the pipette straight up and down when you're drawing up liquid. Do, change your tip for every single new sample, no exceptions. Do use the right color-coded tips. It's an easy sanity check. And do store it upright on a stand. Now for the don'ts. Don't ever try to dial a volume outside its listed range. You will break it. Don't lay a pipette on its side if there's liquid in the tip, and don't ever dip the main barrel into your liquid. These are simple rules, but they make a huge difference.
And finally, let's talk about taking care of your tools for the long haul. A micropipette isn't just a piece of plastic. It's a precision instrument. And honestly, treating it with respect through good maintenance is a huge part of being a good scientist.
And good maintenance is actually pretty easy. Keep it stored upright on a stand. Give the outside a wipe with 70% alcohol now and then. Check the seals and O-rings every once in a while to see if they're worn out. And this is a big one. Never, ever use a lubricant that isn't approved by the manufacturer. You could do some serious damage to the inside.
And that brings us to calibration. Look, checking your pipehead's accuracy every 6 months or so isn't just a nice to do. It's absolutely essential. It's the only way you can be sure that when the display says 100 microliters, you're actually dispensing 100 microliters. Because without that check, all of your perfect technique could be for nothing if the tool itself is off. You're basically putting all your results in jeopardy.
So, at the end of the day, what's the big takeaway? The micropipette is way more than just a tool. It's a symbol of your commitment to accuracy. Getting this right is one of the most fundamental skills in the lab, because in science, every single microliter matters. Precision really is the foundation of everything we discover.